So in the last segment of this week, we will conclude by some examples, and I'll start off with something very basic. For those of you in biological sciences, you may be familiar with these graphs here. These are gels. What is happening here is that there are proteins that are put at the end of the gel, and then based on a, for example, electrical gradient, the proteins start to wander in one direction in this gel. And the proteins carry radioactive charge like phosphorus-32, tritium, or carbon-14, and where then they are exposed to a camera, and we can see how far the protein has moved. And this is a way, an imaging way, to determine the mass of a protein of a product that one has isolated. The procedure that's used here is we have the support here, we have the specimen, in this case it would be the gel, and on top of here is the photographic emulsion, that is the electrons that pass into the photographic emulsion. They create a chemical reaction, which is what essentially is imaged here. So this is a standard way of producing early images, and this has been used also mainly some time ago for imaging, for example, brain slices. So what was done here, this is a rodent brain cut into slices. These slices are then placed on a support, and the film is placed on top of it. That creates in the photographic image here of the radioactivity that's inside this brain slice. So obviously there was some radioactivity injected into this animal here, and we can see here some compound that's accumulated in this particular area implicated in Parkinson's disease, namely substantia nigra. On these examples, I want to end with a very prominent example from monkey brain. So this is now uptake here in the brain of glucose, which is a main substrate that allows us to think. Here's the glucose, radioactively marked. This is the fine structure of the area. And now what this monkey, so this is a brain slice, so this monkey was exposed to radioactive glucose and to some task. The task we can see here. The monkey was looking at this image here. When looking at this image, there's been a representation of glucose uptake in his visual cortex according to the activity of what he's seeing. And actually what one can see here is the uptake of glucose is high in a pattern that resembles to the structure that he has been looking at. So we actually have this has been a very important insight, is we have a representation with imaging, a representation of the outside world directly in our brain. That is a fairly good match. And a few years back, some researchers in Boston have actually shown with functional MRI, showing them the letter M as in Massachusetts, that this letter M showed up also in the brain of the volunteers. So why would we even bother developing biomedical imaging techniques? What is the advantage compared to tissue analysis? The examples that I just gave you actually don't give us any advantages, major advantages. Well, I'll give you an example here, and we are looking at mice that were subjected to 30 minutes of stroke, and they were assessed using MRI at 3 hours, that's here, 7 hours, before, 3 hours, 7 hours, and 24 hours after the stroke. This is the same mouse that we are looking at before and after the stroke at different time points. In the end, after 24 hours, the brain was extracted, slices were cut, and with a staining, Nissl staining here, the neurons were counted. One can see very nicely the lesion of the stroke here, corresponding to what we see in the MRI image. So we have one mouse here, we've looked at it over 24 hours, and in the end we can do a tissue analysis. An extraction. The tissue extraction is pretty much always final if we want to look at the tissue, and there is no continuation of the study. So we have the ability to produce longitudinal studies and repeated studies. So let's say if we take imaging, bioimaging, relative to histology or invasive tissue analysis, typically the information is gathered relatively rapidly. We acquire these images in a few minutes. They were acquired in a few minutes. Tissue dissection means freezing the tissue, dissecting with a microtome, imaging, we are non-destructive. The mouse, in principle, after this measurement and so on, it was the same mouse, so not impaired by the fact that we took the image. So one has minimal perturbation. The bioimaging techniques are either in situ or even in vivo, typically in vivo. And therefore, repetitive, that is longitudinal studies, are possible, which gives us an increased statistical power, a reduced number of samples therefore, and these are all desired features. Here's an example of the ultrasound of a mouse heart. We don't think of ultrasound as a technique used on mice and certainly not for mouse heart. Think about it, how small a mouse is. Think about how even smaller the mouse heart is. One can develop a technique, the ultrasound technique here, that is adapted to such small geometries. And as shown in this video, one can obtain very good images of the beating mouse heart in real time. Think about it, how fast a mouse beats. It is several hundred times per minute to get an appreciation of the challenges that were overcome here in this example. And of course, we can also see here the volunteer, the mouse, which all it is subjected to is putting a transducer on its chest. So we have some other examples here for biomedical imaging. What you see here on the left is a 3D rendering of a tumor. You can see the image. So this is an image taken. It's an MRI image taken of a person for surgical planning. You see on the top of the head the fiduciary markers that allow the neurosurgeon to determine exactly the stereotaxial, that is the coordinates of the brain. And then with the contrast that's generated by the image, with a computer, a three-dimensional image is generated. Based on the contrast, color-coded, we have the tumor highlighted, as you can see here, and that tumor is then very well defined in the image with respect to the fiduciary markers in its position, and very often it's much easier to distinguish the contrast, that is to distinguish the defective tissue, the diseased tissue from the normal tissue, than looking at it by eye. So this helps enormously in either planning where exactly to do the surgery, or if you do radiotherapy, to have an exact location of the tumor. Here's another example of a very often used technique, that's positron emission tomography, and it illustrates its major use in imaging. You can see here this is glucose. You see the brain takes up a lot of glucose. We use glucose to think. We have also here in the bladder glucose for obvious reasons. The glucose that's injected ends up in the bladder, and this part here in the center is the heart. Next to the heart, you can see this dark spot that shouldn't be there, and that is a metastasis. So with this technique, where it's used in practical means, is to detect metastases, determine how many metastases are there over the whole body, and this is of course important for deciding on what treatment approaches to use. And here's the last example. This is functional imaging of a complex task. I think it was a mental rotation task in this case, showing the ventral medial stream. And in such a fairly complex mental task, one can see the many brain areas that are activated. They are shown in red, overlaid on an anatomical image of this subject's head, obtained with MRI. So this technique, functional MRI, provides an insight into the activation patterns when one does a certain task, how the different brain areas are involved. So these are just some examples to illustrate some of the potentials, some of the applications of the techniques. We will do during the course. We will, of course, illustrate every technique that we discuss with examples typically at the beginning, at the end of the lecture.